Okay. So hi, my name is Nathan. I'm a junior biology major, or correction, biochemistry major at Baylor University, and I'm in the clinical physiology lab under the direction of Dr. Takis, where we are developing a in vitro model of ischemia reperfusion to study mitochondrial function for PAD, or peripheral artery disease. So peripheral artery disease, or more commonly known as PAD, is a group of diseases which affect the arteries other than those supplying blood to the heart or brain. And the most common cause of PAD is, in fact, atherosclerosis. PAD is often associated with an increase in cardiovascular burden as well as exercise limitations. And even those patients who are asymptomatic experience severe reductions in exercise performance, which is often measured through walking tests and other things similar to that. The pathophysiology of the functional impairment associated with PAD is believed to be linked to oxidative stress, which is caused by cycles of ischemia reperfusion. So, ischemia reperfusion is, in essence, the cycling between a normal level of oxygen that's being supplied to the tissues, followed by ischemia, or a cutoff of the blood supply, which leads to a lack of oxygen, which is referred to as hypoxia. And then following rest or relaxing, a large amount of blood then goes back to the tissue, which brings an abundance of oxygen to restore the balance, which is referred to as hyperoxia. So in PAD patients, limb ischemia is caused by exercising or other physical movement, which leads to this lack of oxygen, which also causes an accumulation of oxidative phosphorylation precursors and other reducing equivalents, which allows electrons to be more available for reduction. And then upon rest, once they are sitting down or anything similar to that, oxygen floods back in, which is accompanied by an increase in reactive oxygen species, or ROS, and oxidative stress to the cells. And in combination, this whole process is known as ischemia reperfusion injury. So our goal was to develop an in vitro model of ischemia reperfusion so we can test therapeutics and other things for PAD. And in order to do so, we use something called an oxycycler, which is able to control the oxygen and carbon dioxide concentrations electronically of the incubator for which we're culturing the cells in. And in order to do this, in order to make the model, we in essence created these cycles of ischemia reperfusion. So we initially cultured the cells, which are primary human arterial endothelial cells, at normoxia, which consists of 20% oxygen, which is the normal standard oxygen concentration for cell culture. And then we proceeded with a hypoxia stage, which consisted of 5% oxygen, and a hyperoxia stage, which consisted of 30% oxygen. And we cycle these in a constant amount of time, so 90 minutes for normoxia, 60 minutes for hypoxia, and 30 minutes for hyperoxia, to create these normoxia, hypoxia, hyperoxia cycles, so we can replicate what is actually occurring in the patients. So what allowed us to come to the determinations of using 5% oxygen and 30% oxygen was, in fact, looking at the oxygen saturation data of skeletal muscle in PAD patients when they were doing something that's called a 6-minute walk test, where they're in essence walking for 6 minutes, and we were able to observe the oxygen saturation with a MOXIE sensor, which is a near infrared spectroscopy device. So this allowed us to see that, as you can see here in this graph, that when they're walking, their oxygen levels decrease to about 25%. So you have a 75% reduction in oxygen. And then upon rest, their oxygen levels shoot back up to about 150% of the normal level. And then you can see here we had 75% reduction for hypoxia, which when you combine that with the 20%, which is what the normal which is normoxia, what is normally cultured under, you'll get 5% O2. And then for hyperoxia, since you have a 50% increase, this will correspond to a 30% level of oxygen for the cell culture. And this is how we were able to determine what percentages of oxygen we were going to culture the cells under in order to create an effective model of ischemia reperfusion. So to measure the actual viability and functionality of this model, we use something called an Ouroboros, which is able to measure oxygen concentration, oxygen flux, along with ROS production through the use of Amplex Red, which is a fluorescent molecule upon the creation of hydrogen peroxide will fluoresce, and then we can measure that with the Ouroboros machine. So we exposed the human aortic endothelial cells to the cycles of normoxia, hypoxia, and then normoxia, hypoxia, hyperoxia, and the oxycycler. And then we were able to then put them into the Ouroboros and measure the mitochondrial respiration and the hydrogen peroxide reduction using Amplex Red. So this graph right here, or this image, is what we see as a readout on the Ouroboros machine. And the blue line here represents oxygen concentration, and the red line is the negative slope of the oxygen concentration, and it represents oxygen flux. On the lower portion, the black line represents fluorescent intensity of the Amplex Red, and the green line is the fluorescent flux, which is also the negative slope of the black line. So this allows us to see how the oxygen changes in consumption concentration for each complex, and how the ROS production changes also for each complex. And we were able to isolate each individual complex by adding these different substrates. So malate glutamate is added to feed complex 1 of the electron transport chain. ADP is added to achieve maximal respiration in complex 1. Digitonin is added to permeabilize the cells. And then succinate is added to feed complex 2. Rotenone is added to inhibit complex 1 in order to prevent the backflow of electrons along the electron transport chain. Cytochrome C is added in order to serve as an indicator of overall cell integrity and mitochondrial integrity to see if they're damaged, since cytochrome C shouldn't, in theory, affect respiration. But if it does, we're able to know the extent of the damage to the cells and mitochondria. Antimyosin A inhibits complex 3, and ascorbate TMPD feeds complex 4. So this allows us to identify the levels of ROS production for each cell, or for each complex of the electron transport chain, which allows us to see how the model is actually doing in replicating PAD, since we know several things that how this relates to PAD. And in this diagram, you can see how, or this diagram really shows the different complexes, and again, how different compounds affect and feed and inhibit the different complexes. So for the actual data we received, this is looking at the ROS production, or the hydrogen peroxide production of the cells. And you can see that the big thing is that your dark blue here, endothelial cells, normoxia, which is the first line in all these things, is relatively low. So you're not getting a lot of ROS production. But when you look at, for example, endothelial cells cultured under normoxia, hypoxia for 3 days, you can see there's a large increase in ROS production, so hydrogen peroxide production. And you can also see that again with the other normoxia, hypoxia for 5 days, and then the normoxia, hypoxia, hyperoxia for 1 day, 3 days, and 5 days. So you can see these large spikes in ROS production compared to the control, which is the normoxia. And this helps us to show that the model is working, and that, like there is in the patients, there is a large increase in ROS species following the cycles of normoxia-hypoxia and normoxia-hypoxia, which also allows us to gain a greater understanding of how ROS levels are affected in PAD patients who go through these cycles every day of their lives. And then this diagram is looking at oxygen flux, so oxygen consumption by the different complexes. So again, you can see that for the normoxia of endothelial cells, the control, oxygen consumption is high for most of the complexes. And then you can see that when the cells are cultured under the different conditions, normoxia-hypoxia, and then normoxia-hypoxia-hyperoxia for the different amount of times, that the oxygen consumption rates are far lower, which helps us to see that this is consistent with mitochondrial dysfunction and that the model is providing an effective way for measuring and demonstrating mitochondrial dysfunction. Okay, so discussion and future work. So this shows that this could be a potentially good in vitro model of PAD for the testing of potential future therapeutics. And it also serves to reduce the cost and time associated with clinical studies or animal studies, which would normally be required for investigating PAD therapeutics and other things. And in the future, we are working to test potential therapeutics for PAD with projects in microRNAs and other things such as hydrogen sulfide donors and other compounds which have been known to modulate mitochondrial function and ROS production. And I would like to thank Dr. Katakis and Dr. Ismail, our postdoctoral researcher who I work with a lot, for all their work and all of the NIH funding and grants that we have received to help make this possible. So with that, are there any questions?
Amir, why don't you go first this time?
Thank you for the presentation. I have a question about the fluorescence assay. Could you explain more? How does it work?
Yes. So the Ouroboros, we use Amplex Red, which upon the introduction of hydrogen peroxide will cleave and changes and is able to produce light, which we can measure via a fluorescent probe in the Ouroboros machine, which allows us to get an indication of the amount of hydrogen peroxide that's being produced by the different complexes in the electron transport chain of the cells we insert into the chambers of the machine. And then we can measure that, as I shown here, in the total concentration and then the flux of that, which gives us a greater understanding of the amount that is produced.
Sure. Thank you. And one more question. Have you ever thought about rescuing the mitochondrial function? For example, when you're doing this ROS production, if you add scavengers, you can just remove this ROS and see how this affects about this NH and NHH production in your cell.
So are you asking about like adding compounds to like kind of restore the function of mitochondria and like protect against ROS damage?
Exactly.
Yes, that is what we're doing right now. We are working with sulforaphane as one of our projects, which is a hydrogen sulfide donor, which will upregulate NERV2 and other elements associated with the anti-inflammatory response element of the cells. And that is what we are looking at right now and developing this model for us to test like sulforaphane, other H2S donors such as allicin possibly. And then another big thing we were looking at is microRNA 210 and how that the introduction of that will be able to affect ROS production and the overall integrity of the mitochondria, and potentially restoring function and protecting them from these cycles.
Good job. Thank you.
Thank you.
Nathan, towards the beginning, you mentioned that the 20%, and then what, 1%, then 30%, or 20 to 5 to 30 was meant to replicate what was experienced in the tissue. But is the tissue starting out at 20%? I don't, it seems kind of high. What is the tissue starting out as?
So the 20% represents the standard cell culture oxygen level that they use for endothelial cells.
Mm-hmm.
So the way my PI, Dr. Katakis, has set it up is that it replicates the percent change that's measured in the patients. So like when they go up to 150% oxygen level, we'll increase the 20% by 50% to match, to come up to 30%. And then when they experience a 75% reduction to of oxygen levels, that gets reduced to 5%. So it's basically a ratio of what is normal for the cell culture of endothelial cells to what's experienced in the patients.
Do you think it would work if you tried to match more directly what the oxygen exposure is in patients? I know the cells don't, the cells are used to getting 20% oxygen in the atmosphere, but
yeah,
not necessarily in our tissues.
I think that'd be a great idea to test, and it should be easy to do with the oxycycler. We can just put in like different parameters, and it should be very easy to test that, and it'd be very interesting to look at that further. And I'm sure we could see how that changes the ROS production and function of the mitochondria compared to this model. And I'll tell them that. It'll be like an interesting thing to look at.
All right, thank you, Nathan. Judges, do you need any more time to finish writing your feedback before we move on? Okay, perfect. Our next presenter is Sanju from Baylor University. You can begin screen sharing and begin your presentation when you're ready.
Awesome. Thank you, guys. Alrighty. So good morning, everyone. My name is Sanju. I'm from Baylor University, and I'm a sophomore biochem major. And today I am going to be talking about the effects of dietary natural products on the fatty acid accumulation in the human liver cell line, specifically the HEPA-RG line. I'll go into more detail on what the HEPA-RG line is. I'm currently working under Dr. Lovado's lab, which is an environmental science lab. My project focuses more on toxicology and pharmaceutical effects. And this is an ongoing project, and I'll talk about the phases that we're working on. Currently, we've just finished phase 1 last week, and we're working on phase 2 now, but we can go into more detail on that. So what is NAFLD? NAFLD is non-alcoholic fatty liver disease. It is caused by accumulation of small droplets of fat in cells. So if you look at the image over here, a healthy liver does tend to have fat droplets. However, due to obesity or type 1 or type 2 diabetes, the accumulation of fat increases, which causes morphological changes in the cell shape itself. And this could lead to cirrhosis and tumorous outgrowths, which unfortunately, if not taken care of, will need liver transplantation. And a lot of the patients unfortunately do die because they go undiagnosed or untreated. And if you look at the NASH, which is the next step of NAFLD, after that point, it's really irreversible. So our target is to not find a medication, because there are plenty of medications in the market, to take care of NASH patients, but to find natural compounds that we could include in our daily diet that could reverse the effects of NAFLD. So I've included a couple graphs here to highlight the need for strategic therapeutic options. On the top right, we have a graph that depicts the US Armed Forces specifically. I chose this graph because I thought it showed in a very controlled environment the increase of NAFLD over a period of 17 years. And this isn't limited to any race or ethnicity, but the numbers keep increasing, and they are projected to increase by 21% in 2030. And this study was written in 2017, and we're already 4 years past that. The death caused by NAFLD is also projected to rise despite the medication. And the graph on the bottom left over here is from the UK. So this isn't limited to the US, but this is a global problem that we're facing, is the rise of NAFLD. This is not liver disease caused by alcohol, but caused by fat accumulation in the cells. So in our lab, we use vitro models, and these can be defined, I guess, by the 3 R's: replace, reduce, and refine. They replace animals, so we're using cells to work with. They reduce the number of animals that we need because you can just use as many trials as you need, and they are refined. And so with the advantage that we have with using vitro models is that you can control your cells. Unlike animals, there's a lot of variation. The amount of control that you get with a 96-well plate is much greater. The specific cell line that we decided to use is HEPA-RG. This is a well-established cell line. It has been used since 2002, so we're talking about 20 years now that it's been around. And these cell lines are able to produce hepatic progenitor cells, and progenitor cells are basically a more advanced form of stem cells. Stem cells can differentiate into virtually any cell in the body, whereas progenitor cells are defined by their ability to differentiate into their specific tissue cells. And once these cells are grown, so I would grow them in T75 flasks, and then after they are grown confluent, I would add differentiation media, which would include some DMSO, which would allow for them to completely mature into human hepatocytes. So our hypothesis is that we could use natural dietary compounds to mitigate the progression of NAFLD and possibly disrupt the fat accumulation in these cells. So the 3 compounds that we've selected are all plant-based, and there has been past literature suggesting these could help with obesity. Nothing has been done in the HEPA-RG cell line before, but the first compound that we decided to test was cannabidiol, which is often known as CBD to us. This is the hemp plant. And then the second one is transresveratrol, and this is found in grapes and red wine. And the third one is indole 3-carbinol, found in Brussels sprouts, broccoli, cabbage. So these are very, again, commonly found compounds. So very easy to include them in your diet if we do find the results that we are looking for. So as I mentioned earlier, there are 2 phases to our study. The first phase is a cytotoxicity phase. And so we're just trying to see if the compounds that we're testing are toxic to the cells at all. We do not want to try to test for therapeutic effects before we see that they are good for the cells or bad for the cells. So we ran an MTT cytotoxicity test from 0.1 millimolar to 0.01 nanomolar, and they were done by serial dilutions. And I have my map on here. Our controls were a solvent control over here. We ran blanks for the top 2 rows and the rightmost rows, and then we used Triton to cause 100% death rate on the far left. The second phase is the pharmaceutical phase, and the more research-intensive phase, I would say. It requires an exposure of oleic acid, which would basically induce the cells to become fat. So you are inducing an NAFLD, you're mimicking the environment of an obese cell, and then we would go ahead and put these compounds into the cells to see if they reverse the effects of obesity. So for my results, so this is again phase 1, and this is the cytotox graph for an MTT assay for cannabidiol. The red line over here depicts the 100% viable. So this 100% was calculated based on the solvent control. And what's interesting here is that these lower concentrations, other than this very high one that did kill about 70% of the cells, these lower concentrations actually cause proliferation of the cell compared to the solvent control. That could indicate that the CBD could be helping the cells grow. Another step we could take was to see the mechanism of action, exactly how this proliferation occurs. But we also calculated, as you can see to the right, an LC50 value to determine if the compounds were toxic or not. An LC50 value depicts the 50% where the concentration at which 50% of the cells are alive and 50% die. So the lower the LC50 value, the more toxic the substance is. That's for CBD, that's for transresveratrol, and that's for indole 3-carbinol. And what is important to note about all three of these graphs and why we did phase 1 is to make sure that all these lower concentrations here is what you would typically consume in a diet, that they are safe for the cell. It is very unlikely that anyone is going to take this dose that is toxic to try to find a therapeutic effect. Whereas here we are now safe to use these compounds as means of therapy. So what does this indicate is that CBD is comparatively the most toxic, but again, as we saw in those graphs, they are safe within that range of concentration used. So future direction would include conduct co-exposures and complete colorimetric and fluorescent testing plates. So we have actually ran fluorescent testing plates with so these are HepRG cells on the left. Their blue circles are the nuclei. And once exposing them to oleic acid, the fat droplets actually accumulate around the nucleus, and you can see that in green. So we are able to qualify this as some environment that mimics NAFLD. And so once we get it to this stage, we would just add the compounds to test if they go back to this or perhaps actually continue their progression of NAFLD. That is for the coming weeks to know. And another thing we'd like to know is predict the metabolic pathway of all these compounds. And these are my sources. Thank you.
Awesome. And now we'll open the floor to the judges first, ask questions, and then our audience members.
Good job, Sanju. My first question is about the selection of these three compounds. You mentioned that there is some data with obesity on the compounds, but there's a lot of potential compounds there. How did y'all narrow it down to these particular three?
Yes, sir. So there are definitely a lot of compounds that have been used. CBD especially has been a very popular use for arthritis and such diseases. And so we decided to just look more into the compound, and we found that they did use in vivo experiments and in vitro experiments, but not with the HepRG. So that was for the CBD. With transresveratrol and indole 3-carbinol, these compounds were also used in similar studies, not for the same purpose that CBD was used, but since they are commonly found, the grapes, broccoli, cabbage, and all these natural produce were found available to the public, we decided that we should test these compounds because they are easy to access.
And are any of these compounds metabolized by the liver into other forms? And if so, does that inform how often you have to replace them into the culture, or did you just do a single exposure?
So we actually did a single exposure. That would be a good idea to study. I did a 48-hour exposure. So I plated them in 96 well plates and then replaced the media without FBS and then dosed them with the various concentrations. And then you incubate them for 48 hours. And then we test to see with an MTT the viability of the cells. So it's just one exposure.
Thank you, Sanju. I have just one question about your phase 2. Do you have any guess what type of metabolic pathway you need to look for?
As of for now, we are assuming that it has to do something with the interaction between the biliary cells and the hepatic cells, but we are not sure exactly what the metabolic pathway is. We've never done this before in the lab. In fact, most of our research is towards environmental science, and as a pre-med student, I decided to do human toxicology. So human cell line, this is the first time, so not yet.
Sure. Thank you.
All right, do we have any other questions from our audience? You can put them in the chat or just unmute yourself and start talking. All right, if not, thank you, Sanju, for your presentation.
Thank you.
Since Andrew is not here, we're going to go ahead and move on to Selena from Tarleton. Once you're ready, you can share your screen and begin your presentation. Thank you.
All right, good morning, everyone. My name is Selena Alvarado. I am a junior biomedical science major and chemistry minor, and I am an intern for the molecular biology and biochemistry lab here at Tarleton State University. So today I will be presenting the modulation of bacterial host phenotypes by Mycobacteriophage PIXI gene products. Antibiotic-resistant infections kill approximately 700,000 people annually around the globe. We are currently using bacteriophages as the possible antibacterial agents. An example of how we are using bacteriophages to combat antibacterial agents is phage therapy. As we can see in this figure below, we can see that tuberculosis is a worldwide problem. By using phage therapy, we can defeat this bacteria and save thousands of lives. So what are bacteriophages? Bacteriophages are a type of virus that only affect bacteria. So bacteriophages will attach to the host cell and replicate their genome within the host cell. Bacteriophages were first discovered in 1950. To the side, we can see a figure of bacteriophage anatomy. So a bacteriophage consists of a capsid that contains the genome. The bacteriophage uses a sheath right here that will deliver the genome down to the spike, where it will then be injected into the host cell. Bacteriophages use a base plate, pins, and long tail fibers to securely attach to the host cell. For our study, we're currently using bacteriophage PIXI, which was found in Houston, Texas. PIXI has a genome length of 61,147 base pairs. It also has 100 genes, and all 100 genes are currently being used for the study. We hope to determine the purpose and function of each gene. And here we can see a micrograph of the bacteriophage PIXI. We can clearly see the capsid and the sheath. We begin our study with molecular cloning and PCR amplification. So all 100 genes from bacteriophage PIXI were amplified via PCR. The PCR products were confirmed by gel electrophoresis using a 0.8% agarose gel and 1x trisphorate EDTA buffer at 100 volts. Here we can see an example of a gel after gel electrophoresis. In this example, we can see that 4 out of the 5 genes examined were successfully amplified. If we see here, we can see that 2 genes have a single band on their lane, and 2 of them have multiple bands on each lane. Gel fragments of the correct size with multiple bands were excised and purified using the Zymogel DNA recovery kit. PCR products that were successfully amplified and only showed a single fragment on the gel were purified using the Zymo Clean and Concentrate kit. For the study, 45 genes were successfully amplified after PCR amplification. We continued to transforming into Escherichia coli. The amplified genes were ligated into the linearized plasmid P extra by isothermal assembly. The P extra plasmids were chemically transformed into Escherichia coli. The transformed E. coli was incubated on LV agar plates supplemented with clindamycin at 37 degrees Celsius for approximately 24 hours. To the side, we can see an overview of the plasmid P extra. Here in this blue arrow, that shows the gene insert. This red arrow here shows Artery, which is a reporter gene that when activated will turn colonies pink. This green arrow here shows an antibiotic immune gene. We continue by transforming into Mycobacterium smegmatis. The plasmids are extracted from E. coli and purified using the Zymo-Zippy plasmid purification kit. The plasmids were then electroporated into electrocompetent M. smegmatis cells. The transformed M. smegmatis incubated on 7-8 agar plates supplemented with clindamycin at 37 degrees for mostly 5 days. M. smegmatis grows at a slower rate than E. coli, therefore it needs to be incubated for a little bit longer. And here we can see an overview of how we conduct electroporation. Here is the plate after electroporation and incubation after 5 days. As we can see, M. smegmatis colonies have a larger diameter than those of E. coli. And another difference that M. smegmatis colonies have to E. coli is that the edges show ovals, so they kind of look like little flowers instead of single dots. We continue to the cytotoxicity assay. The colonies of the transformed M. smegmatis after electroporation are selected and serially diluted using 7H9 media. The dilutions are spotted onto 7H10 clindamycin plates containing various concentrations of ATC, which is an inducer that triggers Pixie function, and then are incubated at 37 degrees Celsius for 2 to 5 days. The changes in colony morphology are then assessed during gene expression. Here we can see an arrangement of how our cytotoxicity assay plate will look. So we use a negative control, P extra O3, a positive control, P extra O2, and 3 different replicates of each colony being used. Here we see the results for cytotoxicity assay. For this example, we will be using gene 32. Gene 32 is of an unknown function, but it is downstream of a lysine B gene, which aids in cell lysis. We can see that we have 3 different plates. We have a non-ATC plate, that means that it has no inducer, a 10 nanogram per milliliter ATC plate, and a 100 nanogram per milliliter ATC plate. The non-ATC plate shows normal growth of M. smegmatis. If we compare that plate to the 10 nanogram per milliliter ATC plate, we don't see a large difference. But if we compare the non-ATC plate to the 100 nanogram per milliliter ATC plate, we can see that colony growth is reduced. We can also see that the colonies turned a pink color. If we recall back to the P extra plasmid, we know that the pink shows that M. cherry is active. Based on these 2 facts, we conclude that the gene 32 is toxic to some degree. We continue to the defense assay. So for the defense assay, 2 colonies of the transformed M. smegmatis are selected and grown in 7H9 liquid media. Aliquots of the cells are pelleted and washed and resuspended with 7H9 liquid media. The resuspended cultures are then plated on top agar overlay on clindamycin treated 7H10 agar plates with and without inducer and without ATC to induce gene expression. So for this assay, we use 3 different bacteriophages. We use Bacteriophage Pixie, D29, and larva. These 3 phages are very closely related. We use these to see if the gene will protect its host from being invaded by another phage. The spotted plates are then incubated at 37 degrees Celsius for 2 days. Here we can see the defense assay results. For this example, we are using PIXI gene 46. Again, this gene is of unknown function, but it is downstream of an immunity repressor gene. We can see that for this assay, we only use 2 different types of plates. We use a non-ATC plate and a 100 nanogram per milliliter ATC plate. If we compare the non-ATC plate to the ATC plate, we can see that all the ATC plates appear pinkish. That is because the mCherry gene is activated, meaning that PIXI is also activated. We can see that the ATC plates express gene 46 while the non-ATC plates did not. In conclusion, 4 potentially cytotoxic genes were identified. 18 genes were screened for superimmunity. We will continue this research by analyzing the PIXI genome, and we will conduct bacterial 2 hybridization assays in order to determine potential gene interactions. This study would not be possible without the support from the Howard Hughes Medical Institute and the Science Education Alliance, the CG program, and Tarleton State University, College of Science and Technology, and the Department of Biological Sciences.
All right, we'll open up the floor for questions from our judges first, and then our audience if we have any.
Very nice job, Selena. I'm not as familiar with the Bacteriophage Genome organization. So could you tell me a little bit more about how you were able to discretely clone out specific genes from their genome?
I don't exactly know the answer to that. We work with the Howard Hughes Medical Institute. They send us primer of each gene, and we continue to replicate them. So every student gets their own genes. They get approximately 5 genes, and we amplify those via PCR, and then we clone those into our plasmid. I don't know why we are working with Bacteriophage PIXI. It was just assigned to us, but I do know that HHMI will send us the gene primers, and we will continue to work with those as we go.
Okay, and so you mentioned this ATC compound. How does that induce PIXI function? How does that work?
I don't exactly know. I just know that it is an inducer, and we use it so it will trigger PIXI function, and we will actually see if our gene is working or not. Okay.
Last question then. I think there was a slide that had a statistical comparison. Yes, so here, would you mind maybe even zooming in on that table and explaining to us the statistics that were used?
Yes, absolutely. All right. So again, we have 3 different types of plates. This one is full of ATC inducer. So like I mentioned, because the colonies are pink, we know that the mCherry reporter protein gene is active. So PIXI function is at its maximum, we could say. And also the growth of colonies is a lot smaller than that of the non-ATC plate. So because of this, we know that the gene itself is reducing function of the bacterium. So we can say that this gene is toxic because it affected the bacterium in a negative way. Here we can see our controls. So P extra O3 is a non-toxic gene, while P extra O2 is toxic. So we compare those with each plate.
Great, thank you.
Thank you, Selena. Just could you simply just go to your slide 8 one more time?
This one?
Yeah. So could you explain one more time this purification that we did?
Yes, of course. So if we go back, after amplified genes are ligated into the plasmid, we transform those into E. coli. Once they are transformed into the E. coli, we extract the E. coli and purify the plasmids. So then the purified plasmids are electroporated into a different microbacterium called Is Nafld. I am not quite sure why we use this specific bacterium, but the way that we electroporated into the Is Nafld is we gather approximately 50 microliters of the bacterial cells, and we mix that with 1 microliter of the purified plasmid. The way that we incorporate that into the bacterium is via electroporation. So with electroporation, we shock those cells and try to make them porous so that the purified plasmid will get into the bacterium. But we try to do this without killing those cells. So if our cells are shocked during electrophoresis, we know that those cells are dead and we are not able to use them for further study. So then after they are successfully electroporated and no cells are shocked, we incubate them on 7H10 agar plates that are supplemented with kinamycin, and we let them in the incubator for approximately 5 days because of slow growth of Is Nafld.
Thank you so much.
All right, do we have any other questions from our audience? You can put them in the chat or just unmute yourself and ask a question. All right, if not, our last presenter is not here, so that was the last presentation. Thank you all to the presenters. You all did a wonderful job. This concludes our Biology 2 division. We also want to thank Dr. Toby and Amir for judging today.